Hello students, very good evening. So welcome to today's class. And before I start my session today, so let me just go through the technicals. And first let me check the YouTube. Yeah, all right. So student, one second. Okay. So today, before we start our topic, let me have a technical checkup. So I'll launch a technical poll. You guys tell me, okay, if the audio and video is clear. So the poll should be on your screen. Please tell me know if the audio, the screen, and the video is clear. So if these three things are perfect, we can move on and we can discuss the rest of the topics. Okay. So YouTube students, you can also comment on the chat box. I'll be here. Okay. All right. So let's get back to the topic. So yesterday C3 and C4 and we understood how the carbon dioxide fixation happens between C3 and C4 plants. So while we discuss about 1 second, yeah, while we hold on, yeah, we also understood the difference between these, right? So finally, what was left out is so let us talk about it. Okay, so we know that there are several factors such as just 1 minute. Yeah. So there are several factors such as we have carbon dioxide. We have light, and then we have temperature, etc. So these are the factors that generally affect the rate of photosynthesis. Now, photosynthesis on a whole is a biochemical reaction, correct, right? So here what is happening? Something is synthesized. So an anabolic reaction happens during the photosynthesis, where using the carbons we construct the glucose molecule. Now, during this process, like this, what do you call the biochemical process, you can see that when more than one factor. Students, can you hear me? I think you can hear me well. Okay. So 1 second. Students, okay. ಈಗ continue. Okay, sorry students. Okay, we'll get back to the session now. So I was saying you that whenever we have more than one factor that is affecting the rate of photosynthesis. Okay, just one more second. Yeah. Mm, yeah, whenever we have more than one factor affecting the rate of photosynthesis, now what happens is whichever shows the least minimum value of change. So whenever it shows the minimum value of difference, so that one will become the limiting factor. Now let's say that we have sunlight or light as the okay, light as the limiting factor. Now, if there is any difference between the nanometers, let's say that we need 680 to 700 nanometers of wavelength, where the photo activity is very high. Correct, right? Now, you just imagine, just for an example, now the value has gone up to 710 nanometer. And if immediately the photosynthesis has dropped within this minute change, now we can consider that light as the very important limiting factor for the photosynthesis. Now, the reason why we are doing this because Blackman suggested that whenever in a biochemical reaction, if we have more than one factor, okay, such as sunlight, temperature, carbon dioxide concentration, and even oxygen concentration when it comes to C3 plants. So if we take all this into considerations, now, what happens? We will understand that we have one value that is coming at a very minimal value, and this value we can call it as a limiting factor. Okay, now you have understood what is limiting factor. Now, let us go and understand each of this factor, how it influences the rate of photosynthesis. Now, first, let us take light as an example. Now, we know that photosynthesis or the entire mechanism depends upon the light itself, correct, right? So now, if you see the 100% of the light that is available for for the photosynthesis or the solar radiation, 100% of it, we observe that is the plant utilize only 10% of the photo radiation, okay, which we call it as PAR radiation or photosynthetically active radiation. Now, this photosynthetically active radiation, what we tell, so this particular radiation is very highly utilized by the plants for the synthesis of food. Now, what happens to this 90% of the sunlight or the radiation that we have? This cannot be utilized by the plants. Okay, so now we understood that out of 100%, we have a very fixed wavelength of the light that is utilized by our plants. Okay, and second thing is intensity of the solar radiation. Now, what happens with the intensity? Now, imagine that we have 12-hour cycle, right? Day and night cycle. In the morning, when the when the light is available for the plants, now what happens as the sun becomes more and more intense? One big factor that the plants have is they lose a lot of water. So now this is called as transpiratory loss. So during transpiration, what happens? The plant will start losing a lot of water because of the opening of the stomata. Now, to prevent this, many of the plants, what they do is during the peak hours of the solar radiation, they shut down their stomata. When they shut down their stomata, now what happens? Now the exchange of gases do not take place. So when the exchange of gases do not take place, now along with the light, okay, it also influences the gaseous intake. Okay, the gases intake will vary. The amount of carbon dioxide available will become very limited. So with the availability of carbon dioxide becoming limited, now what happens? Your lights, I mean, like the photosynthesis will not go at a optimum level. Okay, so this is about light being the limiting factor for the rate of photosynthesis. Understood? Next one is the temperature. Now, temperature is a very important factor. Like that, 1 second, I'll just move this panel. Okay, so just look here. If we talk about temperature as a limiting factor, now even though the plants depend on light for this, for the photosynthesis, now this particular temperature is since this entire thing is a biochemical reaction, temperature becomes a very important factor. So we can observe that many of the cryophytes, they can photosynthesize at minus 35 degrees Celsius. And some of the algae which live in the thermal vents, they can perform the photosynthesis or preparation of food at 75 degrees Celsius. But most of the organisms, they depend upon for 10 to 40 degrees. That is where you see the optimum photosynthesis that is happening. So if you see which is the proper range where the highest photosynthesis is taking place, that is between 25 degrees Celsius to 30 degrees Celsius. Okay. Now, next coming to another factor, that is the carbon dioxide. Now tell me, for the entire photosynthesis, carbon dioxide is very, very important, right? Why? Because we know that the end product of the photosynthesis is to prepare glucose. Now, carbon dioxide is what contributes to the formation of glucose. In our atmosphere, the carbon dioxide volume is very, very less. Okay. And usually what happens is the concentration is increased to 15 to 20%, 20 times during the photosynthesis, understood, within the plant. Now here, higher percentage of concentration of carbon dioxide is toxic to plants, but they can withstand up to a certain level of carbon dioxide concentration. Okay. Now, when you see that the carbon dioxide that is present, how it is utilized between the two different plants, that is C3 plants and the C4 plants, the C3 plants actually become vulnerable to the shortage of carbon dioxide. Since they depend upon the atmospheric carbon dioxide and they need continuous supply of the carbon dioxide from the atmosphere, that is the C3 plants, that is when their net productivity decreases in higher temperature. Whereas if you look at C4 plants, they can withstand a very wide range of temperatures and they can also prepare or regenerate their own carbon dioxide through we saw that Kranz anatomy, how they regenerate the carbon dioxide, right? So that is why they become very, very optimal with respect to the carbon dioxide concentration. Now you can see that the threshold, that the minimum point of the carbon dioxide is less than 10 parts per million for C4 plants. Okay. And you can see that for C3 plants, you need 50 to 100 parts per million for the C3 plants to have optimal photosynthetic output. Okay. Next one is water. Now, water is also very, very important for the photosynthesis because you know that photolysis is one of the important, what do you tell? Photolysis is one of the important reaction that happens during the light reactions of the photosynthesis. So now if you see that 1% of the water that is utilized, okay, utilized 1% of, sorry, plants utilize less than 1% of the water that is they totally absorb for the process of photosynthesis. Rest of the 99% of the water is lost in the form of transpiration. Okay. So water indirectly becomes a factor that affects the rate of photosynthesis. Okay. Now, why water? Because water increases the turgidity. That indeed increases the exposure of leaves to the sunlight. And then it indirectly supports the role of photosynthesis. Next one is oxygen. You know that the oxygen concentration is higher in atmosphere and it also plays an important role when it comes to the rate of photosynthesis. In C3 plants, whenever we have very high concentration of oxygen and then very low concentration of carbon dioxide, we know that there is a process called as photorespiration takes place. What is photorespiration? Now your Rubisco, instead of oxygenase activity, it goes into carboxylase activity. Now when it does that, the Rubisco, instead of converting into 3-phosphoglycerate, it converts into phosphoenol pyruvate. Okay. Now this is useless process and it releases carbon dioxide and also utilizes ATP. So this type of respiration, which is a wasteful process, happens because of the concentration difference of oxygen and carbon dioxide in the C3 plants. That's why we need a little bit lesser oxygen concentration compared to that of carbon dioxide with respect to C3 plants. Now, there are several chemicals which act as inhibitors, okay, for the photosynthesis. So these are very, very important examples because it was asked in one of the NEET examinations. Cyanides, hydroxyamide, H2S, carbon monoxide, iodoacetates. So all these are basically the ones that inhibit or the rate or reduce the rate of photosynthesis. Okay. And there are others such as chloroform and ethers, so which also inhibit the photosynthesis. Okay. Now, herbicides such as dichlorophenol, dimethyl urea, that is DCMU, and then you have CMU, that is chlorophenol dimethyl urea. Very, very important names. That is dichloro dimethyl urea, and then we have chloro dimethyl urea. So, and also we have they're also photosynthetic inhibitors. So they deactivate the photosystem too, thereby inhibiting the Z scheme or the Hill reaction. Okay. So remember the names of this photosynthetic inhibitors. And the availability of minerals such as magnesium, very, very important, chlorine and calcium ions, they all help in photolysis of water. Okay, excuse me. They all help in the photolysis of water. Now look here, magnesium and iron is very, very useful in the synthesis of chlorophyll, whereas the potassium is used as a translocation of solutes. So all these minerals, they play a very important role in photosynthesis. So their presence or availability in optimum levels is always important. Okay, next one. We have hormones. So this is also a very important topic. Hormones such as auxins, gibberellins, cytokinins. So they all increase the rate of photosynthesis, and abscisic acid, they decrease the photo rate of photosynthesis. Remember ABA, that is abscisic acid. Abscisic acid decreases photosynthesis, people, and rest of the things, that is auxins, gibberellins, and cytokinins, they are known to increase photosynthesis. Okay. Now, sir, so this is about some of the factors that affect the rate of photosynthesis. Okay. Now we have to, okay, oh, hold on. We have to talk about another topic. So let me share the PPT. So this topic is new one, that is respiration in plants, which is a very, very important topic as well. So we'll continue talking about this a little bit, and then what we will do is tomorrow we'll solve questions for both the chapters. So that will finish one of the major chapters in plant physiology. Okay, so give me a second. Um, okay, we have to talk about respiration in plants. So 1 second. All right, students. So let's have a quick look at what we have to discuss with respect to respiration in plants. So what is respiration? See, if we learn about respiration in plants, it is similar to that of respiration in animals. So it's a very, very important process that happens where whatever the energy that is synthesized in the form of glucose, or it is formed in the form of starch, fats, proteins, oils, etc. However it is available, our body knows that once we have the source of energy, which is called as a respiratory substrates, what we call it as, like for example, glucose. Now, what happens is this particular substrate, okay, such as glucose is broken down into pyruvate, okay, it releases energy. So this energy is stored in the form of chemical bonds. So this process is called as cellular respiration. Ultimately, the process of cellular respiration is to provide currency of energy such as ATP molecules. Okay. Now, coming to the cellular respiration, how does it happen? So, breathing and respiration are the coordinated, mechanisms of this entire respiration. Whereas in cellular respiration, what happens in the cell? It is an enzymatic reaction, okay, where biological oxidation of food materials takes place within our cells, okay. And what do we use for the oxidation? We use oxygen molecule. So we use oxygen molecules as a combustible substance, and then we oxidize our substances such as glucose, and where we produce carbon dioxide, water as the waste or the byproducts, and then we have energy that is released in small steps during this process of conversion or biological oxidation. So whatever the energy that is released, we're going to convert them into or store them into ATP molecules. Okay. Now, why do we require energy? You know that all the cellular activities like transport, muscle coordination, bioluminescence, okay, chemotherapy, homothermy, locomotion, nerve impulse, conduction, and the growth, cell division, and then immunity, germination, all of them require energy. So the all the source of energy, okay, is enderogonic activities in all living organisms such as plants, and it all comes through oxidation of organic molecules such as glucose. Okay. Now, if we talk about the energy that is released, released by the oxidation of organic molecules, they're very high rich in thermal bonds. So, this thermal bonds of the ATP whichever is produced. So this can be used by the cells for the ready-made work. So once the ATP is formed, the energy can be utilized by our cells for all the energy requiring reactions. Okay. So the process in which the three phosphate groups are removed from the ATP molecules. Okay. So this ATP molecule is an intermediate energy transforming compound between the energy substrate and the biochemical reactions. Now, why do we respire? So, in the respiration process, like what are the steps in respiration? We know that all these metabolic processes require energy. Energy produced, okay, is regulated according to the requirement of all the activities. It can convert the soluble foods into, sorry, insoluble foods into soluble form. Okay. And intermediate products of the cell respiration can be used for different metabolic pathways. Okay. So it liberates carbon dioxide, which is used for the photosynthesis. Krebs cycle is found for the breakdown of carbohydrate, fatty acids, as well as amino acids. It activates different meristematic tissues of plants. So, anyways, let's see the important aspects of the respiration. First, we talk about what we use as a source for respiration. And the first thing is respiratory substrate. So when I talk about as a respiratory substrate, just like our cooking, how do we use wood for the burning? Okay, and making the fire. Similarly, we use some of the substances such as glucose, fats, proteins, etc. as substrate or the base substance through which we create energy. So carbohydrates, example, we can use glucose. Okay, one second. Glucose, fructose, sucrose, starch, insulin, hemicellulose, etc. are all, insulin is not the sugar, okay, carbohydrates. So rest of them are all main substrates which organisms use for the oxidation during respiration. And there are examples of other than carbohydrates. This is the main source of energy, okay? Main and immediate source of energy. Now coming to fats, under certain conditions, especially carbohydrates have been, whenever it gets reserves get exhausted, especially when you're going for a lot of fasting. Now, fat is also used as respiratory substrate. Okay. Now, what is done to the fats? Fats are hydrolyzed. Okay. Hydrolyzed means we know that there is glycerol, and then we have fatty acid. So this glycerol and fatty acid is broken down into two different compounds. So that is called as fat hydrolysis, where it gives rise to fatty acids and glycerol, okay, by an enzyme called as lipase. And this again is used for the conversion of hexose sugar. So that is how it helps in generation of energy. We'll discuss more about this. And we also use protein as a respiratory substrate. So whenever we have absence of carbohydrates and fats, protein also gives very important source of energy. Up to 8% of the energy is produced by the proteins. So when we break down proteins into peptides, peptides into amino acids, and again, amino acids are entering our Krebs cycle for the manufacturing of our ATP molecules. Okay. So these are called as respiratory substrates. What are those? Carbohydrates, fats, and proteins. Okay. Next one. Based on different types of organisms, respiration also depends. So we know that respiration is highly oxygenic process. So it requires oxygen for the breakdown or the combustion or oxidation of the substances. Now, we know that in our living system, we have obligate aerobes like us. We are compulsory, we need the presence of oxygen, otherwise we are going to die. So when we all depend on the oxygen all the time, we are called as obligated. That means compulsory. So here, oxygen is used for our survival or for our respiration. Next, we have facultative anaerobes. So here they respire anaerobically, but they need the presence of oxygen under certain conditions. Okay, they can switch between aerobic and anaerobic. So example is yeast, parasite, and then which is present in the elementary canal. Next one we have is obligate aerobes. So when we talk about obligate aerobes, so they are obligate anaerobes. Like us, how we are completely depending upon the presence of our oxygen. Similarly, they don't want any of the oxygen condition. So here they are major ATP yielding process will be chemosynthetic maybe. Okay. So examples are Clostridium, and then we have Tetni. So they're strict obligate anaerobes. Then we have facultative aerobes. Again, they are primarily anaerobic organisms, but they depend on oxygen for respiration. Okay. Now, when we come about types of respiration. We have anaerobic respiration. So both the aerobic respiration and anaerobic respiration, it starts from the first step, that is the glycolysis, which is an anaerobic process. So the first step of aerobic respiration is also anaerobic respiration. Okay. Now again, based on the availability or the utility of the oxygen, we can divide this into aerobic respiration and anaerobic respiration, right? In anaerobic respiration, sorry, in aerobic respiration, what happens? We use oxygen where it is completely oxidized, used for the oxidization of organic food, okay, or mainly especially sugars, which are broken down into energy or ATP, right? So again, this aerobic process can be divided into glycolysis, and then it leads to the production of pyruvic acid. Okay. So see here, when we start talking about aerobic respiration or anaerobic respiration, let's say we start with aerobic respiration, we should start with a process called as glycolysis. Glycolysis is an anaerobic respiration. So first the glycolysis takes place. Here there is no requirement of oxygen, okay, for this process. The reason is they say that evolutionarily, okay, before there was an oxygenic atmosphere in our Earth, so there was no oxygen at all. So that is why this organisms developed a way to break down the glucose without the use of oxygen. So that is the glycolysis. The glycolysis is also called as EMP pathway because it was discovered by three scientists. One is Emden, Mayeroff, and then we have Parnas in 1930. So first, this is how the glucose metabolism starts. Okay, glycolysis means glyco means glucose, lysis means breakdown. Okay, in this process, what happens? Glucose is a 6-carbon containing compound which has six carbons in its structure, C6H12O6, right? So that means what? It has 6 carbon atoms in its structure. So this six carbon atom, during the process of glycolysis, it is broken down into 3 carbon containing compound called as pyruvate. We'll understand. So this 6 carbon structure is broken down into 3 carbon structure, which is the pyruvic acid, that is called as glycolysis. So where does glycolysis occur? Glycolysis takes place in the cytoplasm, okay, and it does not require oxygen. It is an anaerobic pathway, okay. So it occurs in both aerobic and anaerobic organisms, okay. So now, whatever the interconversion of your glucose into pyruvic acid, it does not happen with one step. It happens with multi-step process. Okay, this multi-step process requires the help of several enzymes coming at right intervals. Entire glycolysis can be steps, each of them with enzyme. Okay. Most are reversible. Only some of them are irreversible. So first, what happens here? First, we have the formation of glucose. So we have glucose, and for the glucose molecule, a phosphate is added. So that converts glucose into glucose 6-phosphate. You can see here in the structure, this is first carbon. 1, 2, 3, 4, 5, 6. 6 carbon atoms are there. If you see in the first carbon atom, you can see that there is a phosphate molecule that is attached. So glucose is converted into glucose 6-phosphate by an enzyme called as hexokinase. So this hexokinase converts glucose into glucose 6-phosphate. Okay. Now the same glucose 6-phosphate is isomerized into fructose 6-phosphate. See, glucose 6-phosphate and fructose 6-phosphate have the same formula. Only isomerization reaction takes place. You can see the difference in the structure, right? So glucose is isomerized into fructose. So this isomerization reaction, what it does, it converts glucose 6-phosphate into fructose 6-phosphate. Once we have fructose 6-phosphate, again we invest one more ATP molecule. See, converting glucose into glucose 6-phosphate, converting glucose into glucose 6-phosphate. Here we used ATP, which was converted into ADP. So whatever the phosphate we got, we add it in the first carbon atom. Similarly, if you want to add one more ATP, one more phosphate, what do? We use another ATP, and then ATP is converted into ADP. Here we get one more phosphate. Here, what happens? Fructose 6-phosphate is converted into fructose 1,6-bisphosphate. That means what? Fructose was having 6 carbon atoms. So in the first carbon atom, there was one phosphate, that was fructose 1 phosphate. And next, what happens is we have 6 carbon atoms. Here in the first position, there was already one phosphate. In the sixth position, we add one more phosphate. So that becomes bisphosphate, fructose 1,6 bisphosphate. So this conversion happens with the expenditure of another ATP molecule. Okay. Now, once this is done, once we have fructose 1,6 bisphosphate, now what happens is here the catabolic reaction takes place where the fructose 1,6 bisphosphate is split into two compounds, each containing 3 carbon atoms. Okay, you can see that the 6 carbon atom compound, that is the fructose 1,6 bisphosphate, one second, fructose 1,6 bisphosphate, which is having 6 carbon atom, is broken down into 3 carbon atom, 2 compounds. What are those? Which is called as glyceraldehyde 3-phosphate. This is very, very important. So the glyceraldehyde 3-phosphate, the 2 molecules of glyceraldehyde 3-phosphate is released because each glyceraldehyde 3-phosphate contains 3 carbon atoms. So each fructose 1,6 bisphosphate contains 6 carbon atoms. So when you break 6 carbon atom into 2 halves, it gives rise to 2 molecules of glyceraldehyde 3-phosphate. Got it, people? So this is very, very important. Now, whatever the glyceraldehyde 6-phosphate we have, again, now what happens here, you can see, give me a second. See here, once we have glyceraldehyde 3-phosphate, the glyceraldehyde 3-phosphate is converted into 1,3-biphosphoglycerate. When it is converted into 1,3-biphosphoglycerate, you can see that NAD plus is reduced to NADH. Okay, so the reduction of the NAD plus into NADH, here the hydrogen atom is removed. That glyceraldehyde 3-phosphate is converted into 1,3-biphosphoglycerate. Now, when the 1,3-biphosphoglycerate is converted into 3-phosphoglycerate, one phosphate molecule is removed. When one phosphate molecule is removed, ADP is converted to ATP. So this is where we get the first ATP as our byproduct and NADPH as the first molecule as the byproduct. Now you can see here. Now we have 3-phosphoglycerate. 3-phosphoglycerate is converted into 2-phosphoglycerate. 2-phosphoglycerate is converted into phosphoenol pyruvate. Phosphoenol pyruvate is converted into pyruvate. So when phosphoenol pyruvate is converted into pyruvate, now again one phospho, that is phosphate, is removed from the structure, again converting ADP into ATP. Here we get the second ATP molecule. So if you observe the glycolysis, here we invest one ATP, here we invest the second ATP. Okay. So in total, what happens is we get only 2 molecules of ATP out of half cycle of glycolysis. Now, why did I tell half cycle? Because we took consideration of only one glyceraldehyde 3-phosphate. Now, if I take one full glycolysis, that means I will get 4 ATP molecules 4 ATP molecules by the end of glycolysis. Understood, students? So this is about glycolysis and how different enzymes help the process of glycolysis to be completed. Okay, just give me a second. Let's see if we have any issues. Okay, so no issues. Perfect. Hello. Yeah, let's continue. So there is no technical errors, I feel. All right. So this is about glycolysis. Now, what we have in glycolysis is very, very important. In glycolysis, we get NADP, NADH, and we get ATP molecules. So whatever the NAD plus and the ATP molecules that we have from the glycolysis, along with our 2 molecules of pyruvate, right? So this pyruvate is very, very important byproduct. So this pyruvate, which is produced in the cytoplasm of the cell, it is transported to mitochondria. In mitochondria, what happens? The pyruvate is again used for the synthesis of ATP. Now, what's the next phase? The pyruvate, you can see here. Okay, just give me a second. Yeah, the pyruvate, what we have, okay, out of the of NAD, so to the pyruvate. Pyruvate dehydrogenase in the cytoplasm itself. Okay, Again, one more molecule of NAD plus is used, where NAD plus is reduced to NADH. Okay. Here, now pyruvate is converted into acetyl. So acetyl forms a bond with CoA. Okay. So when it forms a very high, it's transported. You can see here, you see the coenzyme A gets attached to the acetate, forming acetyl CoA. Now this acetyl CoA is transported to the mitochondrial matrix. Now here, what happens to the fate of acetyl CoA, which is the pyruvic acid that was converted into acetyl CoA? So that is what is represented by Krebs cycle. So after glycolysis, we have to study Krebs cycle. Okay. The next phase of how the energy production takes place. Now, the cycle was discovered by a German biochemist called as England. So he won a Nobel Prize for discovering or enumerating the entire Krebs cycle. Okay. Now, where does the Krebs cycle takes place? Hold on. Where does the Krebs cycle takes place? Excuse me, students. Hold on. Check this. Sorry, students. Sorry for that. Okay, the cursor was stuck. Anyways, now we have to talk about Krebs cycle. So I was talking about the place where the Krebs cycle occurs. So the site of occurrence of the Krebs cycle is in the mitochondrial matrix. Okay, in the mitochondrial matrix, the Krebs cycle occurs. So now you can see that the Krebs cycle consists of 8 steps in it. Okay, so the Krebs cycle takes place through 8 steps. Okay, now what happened? The pyruvate that was produced could not be used directly. So the pyruvate that was produced in the cytoplasm was converted into something called as acetyl CoA. So that we understood, and then we understood the enzyme that is helping for this process. Correct, right? Now, now what we have to understand is what happens in the Krebs cycle. Once we have citrate, hold on, I'll just use the annotation. Okay. So see here, students. So once we have the glucose that was converted into acetyl CoA. Now this acetyl CoA, it takes the help of something called as oxaloacetate. Okay, we have oxaloacetate. So this oxaloacetate gets condensed or gets mixed up or added with your acetyl CoA. Oxaloacetate plus acetyl CoA, they together condense to form something called as citrate. Okay. So this citrate is formed with the help of an enzyme called as citrate synthetase. So citrate is a starting material for the Krebs cycle. To get the starting material, we should already have oxaloacetate. So and then we should have acetyl CoA. Now we understood how we get acetyl CoA. We get acetyl CoA through pyruvic acid. Pyruvic acid is converted into acetyl CoA. Now oxaloacetate plus acetyl CoA will give us what? Will give us citrate. Now, citrate is converted into isocitrate, okay, which is an isomerization reaction. Once we have an isocitrate is converted into alpha ketoglutarate by reducing one molecule of NAD plus to NADH. Now, whatever the byproduct we are getting here as NAD plus and NADH, this is very, very important. The NADH is very, very important for the next step, okay? So now isocitrate is converted into alpha ketoglutarate. Once we have alpha ketoglutarate, again it is converted into succinyl CoA. Now, this is very, very important step again. Why? Because during the process of conversion of alpha ketoglutarate to succinyl CoA, you can see here one molecule of carbon dioxide gets removed. And also, the NAD plus is again reduced to NADH. So, these two important things. Here we get one molecule of NADH. Here we get the second molecule of NADH. All right. Next, what happens here? The alpha ketoglutarate, which was converted into succinyl CoA. Now, the succinyl CoA is again converted into succinate. Now, during this conversion of succinyl CoA into succinate, and a molecule of GDP is converted into GTP, which is very analog, where again the GTP acts like ATP. Okay, eventually one molecule of ATP is formed here. Okay, so next, the succinate, what happens is here you need the regeneration process. Why we need the regeneration process? Because we need the oxaloacetate for the next cycle. So the succinate is converted into fumarate and the fumarate is converted into malate. During the process of succinate into fumarate, now you can see here a molecule of FAD is reduced to FADH2. Okay, and then during the conversion of fumarate to malate and malate to oxaloacetate, again one more molecule of NAD plus is reduced to NADH, where when malate is reduced to oxaloacetic acid. Now we have oxaloacetic acid, which is the primary ingredient for the Krebs cycle to take place. Now, by the end of Krebs cycle, what is happening, people? You can see here one, two, three. Three molecules of NAD plus is reduced to NADH. And then we have one molecule of FAD plus FAD is reduced to FADH2. One molecule of ATP is synthesized during this Krebs cycle. Okay. Now, where does it take place? As I told you, that it takes place in the mitochondria matrix. Okay. Hold on. I'll just. Okay. Got it, students. Okay. So this is about the Krebs cycle. Okay. Let me change the slide. I hope you're understanding. I'll just see if you have any doubts in YouTube. If there are any comments. Okay. No comments. So let's go. Give me a second, students. I think the PPT. Excuse me. Uh, Sorry about it. Yeah. I'll just change my screen. Give me a second. Do we have it? Hold on. Okay, students. All right, students. So just 1 second, okay? Just 1 second. I think the PowerPoint is stuck a little, so I'll just fix this. Okay, I think it's not working.